Okay, so my name is Bodén. I'm currently at the University of Queensland. I thought I'll just show you where that is on the map. And has anyone here been to Australia? So this is where we are. This is the main campus for the University of Queensland. This is where we are, and you can enjoy some of the photos I think that people have put up there on Google Maps. I was quite impressed myself by that. We're getting quite a lot of rain at the moment, I hear, but usually it's quite sunny there. Okay. Now I'm going to talk about sequence motifs. So what do you know about sequence motifs so far in the course? Have you talked about that at all? Yes, Burkhard is nodding.
So. I haven't talked about it, they haven't listened about it.
Okay.
That doesn't answer your question.
Okay, so maybe we should take this as a test. And I'll go through the slides and I'll ask you questions about them. Okay, so you let me know if I'm just talking about things that you already know very well, okay? I'd like to keep this interactive, so just ask questions or make comments, whatever you like. I'm happy to be interrupted whenever. Or say if I'm going too slowly as well, or too quickly, which is unlikely. I think you'll be able to understand most of this material. I'll be talking about a couple of aspects of these sequence motifs, namely how to represent them, to find the appropriate descriptors for short sequence segments. In part to deal with issues such as finding matches to them. So we scan sequences and we find where these motifs occur, and also methods of discovery. So how do we find them in the first place? And I think that discovery is something that we'll deal with on Thursday. But we might be able to go through some of this material more quickly than I anticipate, in which case we can talk about it today. Okay, so this is a data file which I use as an example of what might be our problem scenario. In fact, this is a so-called bed file. Have you seen bed files before? BED. Does it stand for browser extensible something data file? Not sure. Anyway, it's used or referred to quite often by people who work with genome data. So if you're doing next-gen sequencing, you often store your data as a bed file. What does it look like? Well, it looks like this. So the first column is a chromosome identifier. Second column is a location, a genome location on that particular chromosome. So these are big numbers because the chromosomes are long. The third field there is also a location on the chromosome. And the two together are the start and end of something that is being flagged as special. Then there are a number of fields that are optional in a bed file, and some of which will contain information about the statistical evidence or the statistical assessment of that call being made. In fact, these are this is a bed file generated from a so-called ChIP-seq experiment. Where ChIP-seq, have you heard about ChIP-seq before? Chromatin immunoprecipitation followed by sequencing. I'll be coming back to this type of example from time to time during the presentation today and on Thursday. ChIP-seq gives us locations on a genome at which a particular target protein binds. So that might be hundreds, thousands, or tens of thousands of actual locations. It's not going to be perfect or precise. It gives us an idea that here is probably where we'd find a protein binding event. So what we'd like to answer is a question such as, what do these sites look like from a sequence point of view? So I take this file and I extract the sequences. These were human sequences, or from the human genome. So I look up the human reference genome and I find what the sequences are at those locations. And I extract those sequences so we can get a clear idea what kind of problem that we're referring to here. So I'd like to know what kind of sequence that this protein binds to. And I can find that somewhere along this sequence. Let's see, I turned that off earlier. So somewhere in that sequence I'll find a binding site. Somewhere in this sequence I should also find a similar binding site, because I know that the same protein will bind to both there somewhere and there. Okay, but I don't know exactly where. It could be at the end, it could be at the beginning. How do I find that? That's the type of question that we'd like to answer. And if we have methods to do that, that would be wonderful. To people who'd like to understand things such as how transcription factors regulate the expression of genes. Because transcription factors, as you might be aware, is a class of proteins that tend to modulate the expression of genes, and they do so by binding in their vicinity in a particular way, and I'll illustrate that shortly. But this is an example problem. There are plenty of example problems where sequence motifs might be central to us, including things such as finding what sequences that make up active sites in enzymes, what sequences that make up post-translational modification sites, so where we see, say, phosphorylation. So we take a particular kinase, we'd like to understand what kind of sequences it likes to phosphorylate. Of course, having that kind of information would allow us to make an assessment for the whole proteome, just by bioinformatically scanning sequences with that motif. And Tanya would have talked about things such as localization signals in proteins. These are often described as sequence motifs. Or, as my first example tried to do, is to sort of illustrate how regulatory binding sites can be expressed as motifs occurring somewhere in the vicinity of genes. So it's the whole range, a spectrum of different types of problems that we can deal with, or should deal with, when it comes to motifs. Some where the experimental data will be very rich. We might have structural information that will pinpoint exactly what kind of interactions that are core to a particular process that involve a sequence motif, and we can maybe identify the motif from there. But rich information is often sparse in that, or that sounds a bit weird, but when we have lots of information about one particular binding event, that might not allow us to generalize. We need many examples. As most problems in biology, there would be variations, and we need to be able to appreciate those variations in this description that we're using here. Of course, that variation can only come from us looking at large-scale data. Sometimes we have access to partial data that will give us hints on what the sequence motifs look like. Sometimes we rely on statistics alone. So if you're interested in, say, text mining, and you want to understand what words that are being enriched in large corpora of text, you might want to first have an appreciation of what words occur in our text, and then what words that occur just by chance. So you have a background frequency, and then you look for words that come up more often than you would expect by chance. You can do that just by statistics, rather than using some detailed information on where you should be looking for that textual signal. And same thing in biology. So we need methods that will help us to represent, search for, and discover the occurrences of these sequence motifs. And I'll try to be a little bit more specific after going through a couple of examples. And I don't think I need to dwell on protein localization signals. I think you've heard quite a bit about them previously in this course and other courses. This one. But I'll just use two examples here. So this is a schematic here which illustrates a number of different pathways. Proteins are being translocated in the cell, and sometimes that localization will be controlled by the presence of sequence signals. So in the protein that is being translocated, that might be a kind of a zip code or a targeting signal that, for various reasons, that will dictate where it goes in the cell. And one of those targeting signals would be the signal peptide. So we have our messenger RNA being translated on ribosomes into an amino acid sequence. The end terminus of that amino acid sequence, there might be a signal peptide. If it fulfills certain criteria, a signal recognition, a particle will associate with the complex of the ribosome and the protein that is being translocated, and the whole thing will be associated with the endoplasmic reticulum, which is one compartment in the eukaryotic cell, and inserted co-translationally. So during translation, it will actually this sequence will be incorporated into the endoplasmic reticulum, and from there on, it might be staying in the ER, or possibly further translocated into any of the follow-on compartments, or even secreted from the cell. This is therefore known as the secretory pathway. So that signal peptide there is, of course, essential for that to happen. So we can, once we have an understanding what it looks like, we can look for that in our proteome and see what proteins that will be secreted, or at least end up in the secretory pathway. Okay, so there is a usefulness of that, and it doesn't really depend on what follows after that signal peptide. So the translocation or that signal will only be in that part of the sequence. And of course, a whole range of different proteins will have that signal peptide, and a whole range of different functions can be entertained by that group of proteins. So we can't really rely on something like sequence alignment, at least not alone. The other example that I'll use is when our protein is being fully translated on 3 ribosomes and then folded up into a 3-dimensional structure, and then incorporated into a compartment such as the nucleus, just by virtue of an interaction induced by a sequence signal that appears on the surface of the protein, a nuclear localization signal. And it does so through the interaction of a couple of proteins. Now, I won't dwell on this at all. This is a slide about the nuclear localization signal, and here's one of the proteins which the cargo interacts with, namely importin alpha. There is also an importin beta in the human proteome and in many other organisms as well. So this is a kind of a machinery that is being conserved across many species. So importin alpha has the role of being an adapter. So it's actually the one that is responsible for interacting with a cargo. And there is a range of different cargos. Okay, so anything that needs to go into the nucleus should somehow be translocated into the nucleus, and this is the most frequent way, as we know, for that to happen. An important alpha will thus be able to, or should be able to interact with many different proteins. They could be proteins that are going to interact with DNA, and it could be a transcription factor, say, that regulated the expression of genes, or it could be some other, maybe a structural protein that make up some structure inside the nucleus. Could be a whole range of different things that it does in there. So sequence similarity in a general sense is not going to hold up. We can't use sequence alignment as a method to detect this localization, at least not alone. So if we look closer at that important alpha, the adapter protein, which in its turn is interactive in something, the important beta, which is the main machinery for the translocation, the important alpha has this section which is highly conserved, and that's where the signal is being detected. So if you go to the PDB, you'll find a number of different complexes with important alpha resolved, where you can see, well, at least bits and pieces of cargo that associate with this particular section here. And if you look at those cargoes, there seem to be a shared or a common element to them, that there is a section of basic residues that associate with that particular site of the important alpha, and this is referred to as the major site. There is also a minor site, and there are some variants of that sequence or the localization signal that have two parts to it. So it has a slightly weaker interaction at the major site, but then there is a complementary association with the so-called minor site. So there is a monopartite nuclear localization and a bipartite one. And you see that you've seen sequence logos before. So these visualizations of sets of sequences aligned with one another. Have you seen this before? Yes? Do you know how to calculate them? No? Not sure? Okay, so we'll be looking at that, so don't worry about it. And Burkhardt might test you on it later. So here's an illustration of the first problem that I mentioned. So we have transcription of genes. So the red bit here is a gene on the genome. The arrow here is indicating the direction of that gene. So it starts there. When we have transcription of that gene, we have a complex of proteins that associate with that upstream region, consisting in part of the RNA polymerase, which is responsible for transcription. So it generates that RNA molecule. And then you have so-called transcription factors that bind to the DNA, or potentially indirectly to the DNA through the use of cofactors, or together with cofactors on the DNA. Sometimes they depend on one another, so they need to associate with one another in order to bind to DNA. Sometimes they act alone. Sometimes they even bind to other regions on the DNA which are spatially proximal, but not necessarily in terms of sequence. So you might have a binding event over here which is close to where the transcription occurs, but it's way upstream or potentially way downstream of the actual transcription start site. The file that you saw in the beginning were sites like this, where a particular protein, a target protein, was binding. Okay? So obviously that type of file will help us to understand what the sequences look like that a particular transcription factor likes to bind to. Of course, those might look very different from transcription factor to transcription factor. So here's an example of a so-called turn helix transcription factor, which has two helices here that are essential to the interaction with the DNA that you see along here. And so there are two parts of that DNA binding site corresponding to them. So there would be some DNA sequence here that is being preferred by this transcription factor. So once we understand that, you probably see the power of that. We can then start to scan all DNA, or at least DNA, which are likely to be regulatory in some sense, and we can identify the genes that transcription factor is responsible or involved in modulating or regulating in some way. And we can start to unravel a regulatory network. And especially with, in the case of human, we're dealing with thousands of transcription factors. Once we know that binding site for each of them, or what the binding sites look like, because they're not going to just be the one, we can actually see how transcription factors operate together, and when they are present in the cell, how they could regulate the genes being expressed. Okay, so there are basically three different types of, or three different ways in which we construct motifs, and one would be from knowledge, so we have detailed information. One would be through consensus. We make use of a multiple alignment of known sequences that are being involved in the process of interest, or it could be through automated discovery. And the latter here would be perhaps based on statistical analysis or machine learning. We deal with things such as overrepresentation. Statistically speaking, we'd like to identify the patterns that occur more often than we would expect by chance. So we look for statistical support, and then maybe identify the patterns that sort of come up more often than they should. Or we might have additional information such as a label with our data points, and we can use machine learning to deal with that. And I assume that you've been subjected to some machine learning ideas in the past here. Is that right? If you don't say no, I'll just assume that you mean yes. Okay, so here's another transcription factor, DNA interaction that have been resolved structurally. And of course this could contain, well, will contain detailed information what the DNA sequence is, but maybe it doesn't hold up in general. These patterns tend to be quite degenerate in that the transcription factor might bind even though we mutate some of those nucleotides. Okay? So we'd like to know what are the critical positions in such a binding motif, and maybe describe that in some way that would be sensible for us to score it, to quantify this across a whole genome sequence. Most of the time we end up with a much less specific data set, but at least one with more data points. This is an example data set from so-called SELEX experiment. This has been around for quite some time. We're still talking about transcription factor binding here, but this is an in vitro experiment. What it does is that it generates nucleotide sequences that this transcription factor prefers. But it's done in a semi-random kind of way. So we end up with a whole lot of sequences, and although we know that they all bind to the transcription factor, we don't know exactly where. So when we look at this, it's kind of difficult to find what that motif should look like. So this is similar to the problem that I mentioned in the beginning, but not quite as hard. Indeed, these are short enough so that we can use sequence alignment. So there is enough sequence similarity at the core of these sequences so that we can use sequence alignment. And in this case, it's an ungapped alignment. And I should say that gaps do not are not as applicable to problems at the DNA level as they are when we're dealing with protein sequences. Especially for transcription factor binding sites, tend to be very little variation in terms of the length, or there are seldom spacers or linkers between parts of DNA binding site. Although there are some examples, but usually they're quite fixed, and we can just have a section in the middle of an alignment which would be less definitive in terms of what letters should occur there. In this case, so this is the transcription factor GATA3, we're actually seeing a shared pattern in the center of this alignment here. So this is in 2 columns here, but it's actually the same alignment. So what do we do with this? Well, we obviously like to quantify things, otherwise we can't objectively make an assessment across big data sets if we use it for scanning. So the first thing that we'd consider would be to count the different nucleic acids that we see in each of the columns of the alignment. So if we take one example here, say column 10, we would count as many as 17 A's, 11 C's, 15 G's, and 13 T's. And that in its turn can be converted into probabilities or frequencies. Okay? And this is something that we could potentially use as part of a scoring method now. That particular column is not very informative though. We don't seem to have a clear pattern there. And in fact, the more conserved part of that alignment would be beyond column 16. It's actually at column 17. So now I switch my attention to this visualization here, which is the same type of visualization that we saw previously with the nuclear localization signal. Okay, so over the next couple of slides, I'll talk a little bit more about this sequence log and how we can generate that, because visualizing these sequence patterns will help us to understand, well, obviously what they look like, but also to convey that kind of information to the biologist. Biologists like sequence logos, but more about that in a minute. First, I'd like to just discuss one choice that I did here, which is not completely straightforward. You may notice that if we look in the first column here, I had actually the same number of counts as for any of the other columns. And if you look at this alignment, you'll see that in that first column, I actually only have one A there. And then I have to go all the way down there to find the second occurrence. So that's a C there. So if I were to count them as the only symbols that occur in the first column, I would end up with a count of 1 for those two and then 0 there. So if I were to convert that into the frequencies, it would be 0.5, 0.5, 0, 0. Can you give me a reason why that might not be what we want? Yeah?
It would be really big in the sequence logo because the information is high.
It wouldn't just be big in the sequence log, it would be a problem for us if we were to use those frequencies as part of the scoring in general, because we'd require, right, that there be an A or a C in that particular position. And we've already seen that, well, there are only 2 sequences in that big alignment that would support that. And it's quite likely that that part wouldn't even be an essential component of what is required for GATA3 to bind there. So it's probably making too much of just a few data points. So that's why we might want to sort of trick our way out of that situation. So if we want to use that column as part of a scoring matrix, we should probably try to use the gap, or the information that the gap provides, if any, at least that we would have relatively little data on which we can sort of draw any conclusions about what symbol actually occurs or need to occur there. So this would be a way whereby we count the gaps as a wild card. That is, that it could be any symbol. But it's just a trick. It's not a great way, but it's sometimes necessary to make these more pragmatic choices. Yeah, Max?
It looks like you have only corrected for the occurrence of A and C, which is why the relative frequency is a little higher than for G and T, right? How about the actual background of...
We'll come there.
Okay.
Yep. It's a very good point. It's just that I'll get there shortly. So frequencies are quite easy to compute. Okay, so it's just a number of the symbols, in this case here indexed with A, for a particular position divided by the total number of sequences. So, for instance, in this alignment here, which is simpler than the one that we had previously, we have 5 A's, and we have 20 sequences in total, which means that that particular element there would be 0.25. And the number of A's that we see in the second column would be just the 2 there at the top. So 2 out of 20 would be 10%. So that's how we determine this whole matrix here. Okay? So that visualization, how do we come up with that? There are a couple of parts to it. The first one being that it's based on information. So the middle expression here is quite important. That gives us the total height of all the letters that can occur in a particular position. So in this case here, we have just T's. So if we go back to this alignment, we only see T's in the third column there. Okay, so we have complete information, if you like. This is a calculation that is based on entropy. How many knows about entropy here? Shannon entropy. Surely you must have come across that previously. What entropy is, if I just go to the definition that I found on Wikipedia, entropy is a measure of the information contained in a message as opposed to the portion of the message that is predictable by inherent structures. So if it's completely random, there is a lot of information in that message because we can't predict it. Whereas if we're dealing with messages like this, it's quite predictable. You probably come up with a guess quite easily. So that's the intuition behind information entropy, or Shannon entropy, I should say. Okay, does that make sense? So there is a calculation involved here as well. Okay, but we don't have to sort of go through that in detail. If we only have 2 different values, like 0s and 1s, and we look at the proportion of them being, well, if you have only 0s, then our entropy would be 0. If you only have 1s, our entropy would be 0. If we have sort of an even number of 1s and 0s, our entropy would be maximal. Okay, so that's the intuition behind it. So if we look again at this expression here, we have log 2 of A, or the size of A here, and A is the alphabet. So if we are dealing with DNA, the size of the alphabet would be 4. So what's log 2 of 4? We can divide 4 how many times before we get to 1? So 2. That's the maximum height. So if we look at this diagram here, so the y-axis has the unit bits. We have 0 up to 2. That's the maximum value that we can get. That's the maximum height for any one position. So we have 2 there, and then we're going to deduct the entropy at that position. So we look at the letters that we have there, and we can determine what the entropy is and we deduct that from the value 2. So in this case here, t, we only see t's. So it's comparable to the scenario where we only see 1s. So we have 0 entropy, so we deduct nothing from the 2. That's why we get a big letter there. That's the total height of a stack of letters, but there are no other letters than T here, so that's why we only see it. But in this case here we have G, C, and that's probably an A down there, squished. In fact, the total information in that column there is much less. So we have the 2 minus the entropy. In this case, it would be an entropy that would be sort of somewhere along there, right? Because we don't have a perfect or sort of an inherent structure that would be entirely predictable. So we end up with something that is in between, and the total height there is slightly less than 1 bit. Okay, you can do all the calculations if you like, but that's the intuition behind it. And then you divide the total height of that column with the frequency of the letter. And of course they would all add up to 1. So G would be the most frequent, most probable in that column. It's actually occurring in 75% of the sequences in our alignment. Does this make sense? So it gives you a quick idea, first of all, how much information there is in each of the positions, or our sort of confidence of making a prediction. So in this case here in position 8, we don't know a lot. Okay, so that's why the whole thing is squished together. It makes very little sense to make a difference between whether we see, in this case, a C or a T, because we're not confident. Whereas in the ones in the middle here, we're quite confident there is a T. There is a lot of inherent structure there, if you like, in the data. Did I say everything? Yes, I think I did. Okay. So these sequence logos, they are being used for proteins as well. And I'll just use the nuclear localization signal again just to illustrate a few more things about the collection of data and putting together an alignment. So this is the same. How do we go about finding the data on which to construct a nuclear localization motif? Well, it could be done just by a database search. This is an entry from, can you say which database? Do you recognize this sequence or this format?
Except for the probability?
Yes.
Switchboard, it comes from switchboard actually.
Good.
So Unicode copied that.
Okay, and I've identified or highlighted here a couple of the fields that we take a particular interest in. First, we know that this particular protein here, doesn't really matter which, but it's been identified as being compartmentalized inside the nucleus. So it's being found in the nucleus. Could go otherwise or to other locations too, but we know that it's been found in the nucleus. Then we have the sequence signal that has been determined in some kind of way. Most of the time people would have had a suspicion where the sequence signal is. So they might have seen other nuclear localization signals, noticed that there is a section of basic residues, and they look for that section of basic residues because they know that this thing is being localized to the nucleus, and they find one there. And then they might mutate that position by position to identify what are the positions that are being critical to that translocation. Otherwise, they wouldn't get their paper published. So someone has gone through the trouble of identifying this as being critical to the localization into the nucleus. So we can make use of this. We can put together all of the data that has this particular annotation in Uniprot and construct an alignment from there. And this is an alignment just like that. It's just that it's based on a slightly different type of data. This is from a Japanese group that performed some experiments. They generated a random peptide library, and then they looked for the peptides that interacted with the important alpha that we saw earlier. And they sort of put together an alignment from there, and from there you can identify the counts of different amino acids. So here I've listed 7 of them, but there would be 20, and I just picked the few that occurred in this particular alignment here, so we can get an idea what the counts are like. And you can see that the lysines and the arginines are quite frequent in all of these sequences. So we would probably score them highly. So now to Mac's comment earlier, you might be able to guess what that box really contains. So it says 0.06, 0.06, 0.09. They correspond to each of the amino acids there. They would be the background frequencies. Because obviously, if we have a sequence motif with something that is very odd, say a tryptophan or something like that, that would be more valuable for us. It would give us a way of increase the integrity of a match, because of the fact that amino acid is so infrequent, finding it is going to be more of a surprise to us. So we should be weighing in that information somehow into our scoring system. Before sort of talking further about that, I'd just like to point out something that I largely disregarded when it comes to sequence motifs. Regular expressions are quite popular. And the 2 blue boxes down here, they are 2 examples of regular expressions that you can construct by looking in these boxes. And you would have seen regular expressions before? Yeah. So this would be, this first example here would be one that considers 6 positions, 1, 2, 3, 4, 5, 6. And in each of those positions it would nominate the possible amino acids that we should accept for it to be a match. Now RG would actually only cover a majority of these guys, but not all of them. As soon as we start introducing more of those symbols into this regular expression, of course our regular expression will be less specific. That is, that it will sort of flag things that are not going to be real. But at the same time, we want our description to be sensitive. That is, that it should incorporate as many of these as possible. So it should at least accept the ones that we know are positives. But there is that balance between sensitivity and specificity that I'm sure that you must have come across before. So regular expressions are largely seen as a more coarse description for a sequence motif, because they will only give you sort of either a positive outcome or a negative outcome. Whereas the types of scoring systems that we'll devise now will be gradual. They would be on a continuum of scores. So we can put a threshold somewhere if we wanted to differentiate between positives and negatives, but where we put that threshold is really up to us. And we can interpret the score or the magnitude of the score as a proxy for saying something about the confidence we have in the prediction too. Okay, so just to finish off here. So once we've started to weigh in the background, we are in a position to construct what I'll be referring to as a positional or position weight matrix, which looks very much like a substitution matrix. And I'll come back to this later on, what the steps are in calculating this. And here again is a sequence log of, in this case, for this alignment. And again, you can see clearly what the conserved positions are. So there are some weaknesses of the type of scoring systems I've been alluding to, and one is that just plain frequencies are not going to cut it. They fail largely to capture the element of surprise when used as a means to scan sequences. So that's where the background comes in part. It's also a case of alignments not always being obvious, so it's not always trivial to construct an alignment from the data that we have. So think back about the chip-seq data that I was referring to, which had maybe 100 or 200 nucleotides, and we had no idea where in that we would actually find our motif. We can't just rely on sequence alignment because most of that sequence would not sort of align well at all. So our actual the sequence similarity that we see in the motifs or the binding sites would not be detectable through alignment. And it's not at all clear that those sequences would have the kind of relationship that homologous proteins have, for instance. So we need a stronger theoretical framework to talk about scoring, and then subsequently discovering motifs. So I'll now introduce some probability here, and most of which I think you already would have seen in potentially other courses. So a probabilistic model. This is a system that simulates the object or process under consideration. So think the roll of a die is our example model, or the example system that we'd like to simulate. We will find parameters for that to infer or assign different probabilities to possible outcomes. So to talk about that, we need to identify all the possible outcomes. We need to say something about the quality of that outcome, whether it's just a number on a continuous scale or whether it's a discrete thing, such as seeing a 6 on the die. And then we find the parameters. So this would be what would help our model to make predictions, or to sort of encapsulate the process of interest here, to simulate it. And we'll refer to them as theta here, as a group of parameters. As with all sort of probabilities, they need to satisfy some conditions. So a probability always need to be positive. When we look at a discrete number of possible outcomes, then the probabilities that we assign to each of them would always have to add up to 1. And in the case of a continuous variable, we know that the density, the probability density across that continuous space would have to be 1 as well. And then we have inference methods, and which follow the rules of probability. And that can allow us to either directly or indirectly infer things such as what the probability of getting a 1 would be. That it could be 1/6, say. And often we use data in order to find those parameters that would allow us to make predictions like that, or infer things like that. So we need to talk a little bit about probability. These are basic things, so I'm hoping that you would already have seen them. So we have joint probability, which is the probability of x and y, which we write like this. We have conditional probability, such as the probability of x given y. Okay, so we know something about y, and we'd like to understand x in light of that information. So more on this. So a joint probability for x and y specifies the probability of each complete assignment of values for both x and y. And this extends for any number of variables. And I can just point to what I wrote on the board earlier for the class this morning. That here's a case of a joint probability for two variables x and y, and we just assume that those two are Boolean variables, that is, that they can take either the value true or false, which means that we can write this as a table, which is simple to understand. So we would have 4 possible atomic events. So these are 4 possible outcomes, that both of them are true or both of them are false, and then we have the other 2 alternatives when they're different. And we would assign probabilities to each of them. We need to make sure or they will add up to 1. But then, of course, the probabilities could be depending on what we've seen, okay, or the information or expertise that we have. Okay, we can extend this to another variable like z or z, and that would mean that we would have how many entries if that's a Boolean variable as well? How many rows in such a table if we have 3 Boolean variables? Take a guess.
8.
8. That's better than a guess, I'm sure. Okay. We can go the other way. So a joint probability can be marginalized, as it's called. That's when we remove one or more variables from it. So if I wanted to remove, say, y from this joint probability, so I'm just talking about the probability of x, I can do that because there are only 2 possible values of x. So it's either true, which means that I just ignore the value of y here, and I get a probability of 0.4. In this case, a probability of 0.6. Those 2 possible. That's the other aspect of the marginalization. Then perhaps the most interesting expression is the conditional probability, which we can calculate from joint probabilities, like this expression indicates here. So we have the probability of x given y. So we know y, and we can determine that from the joint probability of those 2 events by dividing it by the prior probability of y. But these are all sort of fundamentals of probability theory, and I don't want to dwell too much on that, but we'll be seeing them a little bit later in the context of motif discovery. It also gives us a better understanding of some of the concepts that I'll be talking about shortly. Now, to just finish off that material, a conditional probability, again, can be defined in terms of that expression that we saw on the previous slide. But we can turn those things around a little bit just by algebraic, some algebra, to say that this joint probability is this expression here. Now this thing here is reflexive, whereas this thing is not. So from there we can derive Bayes' rule, and sort of, I'm hoping that you would have seen this before, but a Bayes' rule will allow us to sort of to derive the conditional probability for one ordering of x and y. So in this case, the conditional probability of y given x, from there we can actually determine the conditional probability of x given y, as long as we know the prior probabilities of x and y. It's a very convenient rewrite that we make use of quite frequently in probability theory. Okay, sequence models. We are interested in understanding the occurrence of different letters in the context of sequences when they happen in a series. And this will be more clear to us when we apply it in motif discovery, but for the time being, we'll use a very simplistic example here to illustrate what it means. So observe biological sequences, which could be any type of sequence. It doesn't have to be biological sequences. These could be corpora of text, for instance. They could be thought of as outcomes of random processes. So something would have generated that sequence in the first place. And one good analogy would be the works of Shakespeare. Something had generated the works of Shakespeare, obviously Shakespeare. And we'd like to model that process, okay? Or if we were to reconstruct that, sort of having looked at the works of Shakespeare and we construct a model to regenerate that, that's a clear indication that we've encapsulated the essence of what generated that in the first place. Although there are some caveats to that. It makes sense to model sequences using generative probabilistic models. So this is how we're going to think about this. So we have the monkey typing on the typewriter and perhaps accidentally reproduces the works of Shakespeare. Here we're going to have a monkey typing biological sequences and they happen to be something of biological significance. We'd like to think of that monkey as our model here. So we're going to fine-tune the brain of the monkey so that it does what it generates the sequences that it has seen. And perhaps beyond that. So you can think of this, you can think of a probabilistic sequence model as a machine that randomly generates sequences. And since you've seen some machine learning previously, you probably recognize some of the philosophy behind how we're going to use a training set, so a D. We think that our parameters can be estimated from this training set. And with a big set of these, we expect the measured frequencies to be reasonable estimates of the underlying probability. So if we've seen enough of the works of Shakespeare, that we should be able to sort of extrapolate to the full works of Shakespeare. Even though we haven't seen all of it, we'd like that to happen. And we expect that the data set would contain all the essential information. So a model with parameters theta and a set of data, that's the scenario that we have in mind. A maximum likelihood estimate for theta is the value that maximizes this expression here. So this is the probability of the data given theta. So this is our monkey, or a model, the model parameters. It's there. If it, with a great probability, reproduces our data, that's our aim really. We'd like to maximize that ability. Okay, so let's come back to that. A simple sequence model, this is just to illustrate some of the points that I made. So we're going back to the rolling a die example, but in this case we'll be using a slightly modified die, a tetrahedral one, so one with 4 sides only. So it looks something like this if you render it in 2D. So it would have 4 sides, and you'd be looking at the letter which comes up at the bottom of it, because there are 3 of them that would be sort of visible and 1 that would not be. So if you want to identify one uniquely, you look at the bottom. You roll this die 100 times and you write down the letters that come up or down. The frequencies of the outcomes are the maximum likelihood estimates. It's that simple, right? At least in the very simplistic model that we have in mind here. With these parameters, the model has the best chance of reproducing the training sequence. So let's look at how this could turn out. So this is our sequence, training data, and I start with a C, ends with a C, and then lots of other letters in between. I think it is about 100 symbols long, but I'm not quite sure. We're going to construct a model. This is written in Python here. You don't have to mine the code really. If you wanted to, you can download this code from a URL that I'll give you probably on Thursday. But for the time being, just think of this very abstractly. We're going to construct this model P1, which is a probability distribution. So we'll have 1 parameter for each of the letters in our alphabet. And that's what we're defining here. And then we're going to train it And we're going to do so by just iterating through this whole sequence, and we're going to look at each symbol S in that sequence here. And we're going to observe it. And then we're going to print it so that we can look at the parameters that we've estimated from this data. And this is our model, essentially. So we see that A is 0.21, C is 0.27, and so on. This would be what we then can use to generate a new sequence. These parameters, but where do they come from? Well, it all happens here, sort of this line here. So when we do observe, what do you think happens? What's happening inside the method observe? It's just doing counting, right? It's very simple. It's not doing anything fancy at all. It's just a simple, the model is just a distribution. So we're just going to count up the number of C's that we see here. And we're going to divide that number with the number of the total number of symbols. And we get this distribution. And now we're in a position to generate sequences. And if you remember the monkey analogy, we should be able to regenerate the training data. So let's see how we go. So we have this new sequence, sequence 2, that we're going to just concatenate what the distribution generates. So we're reproducing or we're constructing a new sequence. But if you look at it, it's nothing like, and for your convenience, I've just added the same sequence as on the previous slide down here. They're nothing like one another, right? And this one doesn't even start with a C, it starts with an A. It does end with a C, and the second last is a T. That's not quite right either. So lots of differences. But if you look at the distributions, they're essentially the same. So if I did the same thing, I used sequence 2 as training data and observe each of the characters, I would end up with a distribution that looks like this. So the probability of an A is 0.21, just like it was in the first. So we've at least managed to replicate something, but not quite the serial nature of this sequence. So how could we do that? How could we capture the sequence, sort of the transitions between symbols here? Yeah?
With a hidden Markov model, for example.
Yes. So, and how would you design that hidden Markov model? What would it do that would be different?
Well, first all the hidden Markov model would take into account always the previous symbol and decide upon that information the likelihood of the next one. Okay, so from this training data you would make an assessment. So once we've seen a C, there is going to be a probability of seeing an A, C, G, or T. And that is a structure that you would be able to infer from this structure that you can then apply here to reproduce something that would be slightly more similar. What's the disadvantage of that approach in this case? You have to decide how big the memory, so to say, is. Yeah. How many symbols you want to take into account.
Okay, so there is more burden on us as designers to figure out what the actual model looks like internally. So what parameters that we need to sort of somehow consider. Any other disadvantages? Would it mean that we would require more data, for instance? Maybe it's becoming a slightly more difficult problem just because we only have this particular sequence on which to base our predictions. So maybe that's just too little to make any solid predictions, and therefore we have to constrain our model or a model's complexity in order to make reasonable predictions. So there is that sort of balance between model complexity and availability of data that we need to work with. Okay, so I'll come back to this when we talk about motif discovery on Thursday. First, let's see if we can develop a better scheme for scoring sequences. So once we have our motif, or maybe we should rethink a little bit how we construct our motif so that we would get more robust predictions of what actually is a match to our original motif. So I'll be referring to position weight matrices here, and I'll change the notation just slightly. So instead of F, I'll be using Q here, but it has the same two indices. We have a position and a particular symbol. So we're going to look at the symbol at the position, and we're going to find how often it occurs. This data is a particular transcription factor binding motif, which has been constructed in a similar way as we saw previously with the SELEX data. So where we had some examples and then we performed an alignment. We found counts that we can sort of put into a matrix like this. And that matrix is copied here as well. So it's the same one as you saw on the previous page. And then the first step is usually, although not exclusively, is usually to add so-called pseudo-counts. The only difference we see between these two matrices is that I've added 1 to all the cells in the matrix, or a constant value. And this will then form the basis on which we score our sequences. So can you give me a reason why we would want to do that? I mean, we're clearly distorting the actual data here, right? This is no longer representing the pure counts. It's adding something to it. What benefits could this have? Yep.
It could be that we haven't observed something in the data, but which is biologically relevant. So then you have it there at least at a really low probability. Exactly.
Yes. So if you think about the more extreme example, that we don't have 59 sequences that we have aligned, but we only have 5. So you'd have a count of 5 there and then 0, 0, 0. Now, to create a representation out of that few examples will probably be stretching it in terms of us committing to a very extreme probability distribution, because we'd be saying that it always has to be an A in that position, whereas we only have 5 sequences on which to base that inference. Well, maybe we should be a bit more cautious and allow some alternatives to be considered at least. So that's why we add pseudo-counts, and that's a way of inserting a prior belief here. So if you think about when we have 0 counts, we're going to have a belief that we see a certain letter just by virtue of that pseudo-count. So if we add 1 to each of them, we would say, let's assume that in the absence of any evidence that all letters occur equally or with equal probability. Or we could weigh them somewhat, perhaps along the lines that Max suggested earlier, that we use sort of a background frequency. We will not do that here because that background frequency can be incorporated in a slightly different way. So let's leave it as a uniform pseudo-count here. And then we determine the frequencies, just like we did before. So you notice here in the second column that we end up with something that is not quite as extreme, that this would be 100%, but it's 95. And we'd still consider some of those other nucleotides there. And then we're going to consider the background. Now the background is going to be different from organism to organism, especially when we're dealing with DNA sequences. We're going to have some organisms that will have a lot more of one particular nucleic acid than the others. Or there is, well, it should be should be that there is a balance between the A's and the T's versus the C's and the G's, obviously, because of the complementarity of DNA strands. But there is going to be different bias in some organisms. And then, in fact, if we wanted to build an even more sensitive system, we can take into consideration the fact that some parts of the DNA in, say, a human would be much richer in A's and T's than C's and G's. And there is something called CPG islands, I'm not sure whether you've heard about them, but which are more rich in the pairs of CG than they would be expected by chance. And these are typically regulatory regions, but that's a different story. So having that background as a separate piece or as a separate component here makes a lot of sense. And we're going to incorporate it, and we're going to incorporate it in the same way as these things are incorporated into a substitution matrix, which I assume that you've seen before. So you talked about things such as BLOSSOM, say. Yeah. So you're going to compare frequencies here from a foreground set with that of a background set. So in order to do that, we can use something like a ratio, an odds ratio between the foreground frequency of a particular letter at a particular position divided by the frequency of that same symbol, generally speaking. So that's the same thing as taking that value there and dividing it by that value there in order to arrive at this. And because it's above 1, we're going to say, well, we're seeing this more often than we would be expecting by looking at any region. So if we see an A there, we're going to lean towards believing that we have a match. Whether, well, the alternative would be that if we see a C in that position, well, a C is more common in our background than it is in our foreground, so therefore we're leaning towards believing that maybe this is not a match. But this thing here, the distribution of these scores, these odds ratios, would be going from 0 up to infinitely positive, but there would be it would go up around 1. Okay, so that's when we're sort of not sure. So it's not a great distribution to work with, so that's why we often do a log transformation, so we make sure that it's centered around 1, because the log of 1 would be 0. Right? And then we would have negative values. So for that C there, that had a ratio of close to 0, it would be negative. Whereas the A here, which was above 1, you'd be dealing with a positive value. So we can look at it as just as we do with the substitution matrices. Okay. And of course, the added benefit of having a log odds score is that we can sum them. So here's an example where we scan a sequence. So this at the bottom here is a sequence that where we'd like to identify whether we have a match or not. Note that it doesn't really matter that whether we're dealing with DNA or RNA or amino acids here. It's just a different alphabet, and of course that those frequencies would have to be found in the appropriate way. So we end up with a matrix, a PWM, or a position weight matrix that looks like this with the log odds scores, and this will stay the same, and we're going to use this as a kind of a window to scan this sequence here. The only thing that will change between the next few slides here is which of these cells that are being highlighted. So we had, in this case here, the first symbol is a C, so it's minus 2.5. Okay. The G is more highlighted here because it's the second letter, it's minus 4, and so on. All the way to the fifth letter, And we add these highlighted numbers together and we get -12. So do we think that that is a match or not? Probably no. Okay, it's a negative number. Here's the next one. So same thing, same matrix, so I'm flicking back and forth between those two. The only thing that changes is which of those cells that are being highlighted, and we're going to highlight the ones that correspond to the letter for each of the positions here. So we have a T there, A, T, and T. And we just add them together, so -11, so that's probably not a hit either. And then we have something that turns positive at least. So in this case here, we have the word tata. Okay? So if I go back to the alignment, or look at the sequence logger here, it's not too far off from this. At least atta will be the same. It's just that the first T here that is not quite clear. But it's positive according to this scoring here. And of course, now we have the option, having a continuous score to deal with, to sort of set that threshold according to our preference for sensitivity and specificity. And we would be able to work out what are the hits in that sequence that we're scanning. And in this case here, this is just a histogram that illustrates the same scores. I didn't mention, but this formula up here is just the log odds scores here, and then we're summing across them. So with log odds, or with log, we're operating in log space. We can just do sums on our ratios here, whereas if we were to not do log, then we would have to do products in order to get the same thing. But logs are useful in that sense, that we can do sums instead of products, and also because the values are more manageable in terms of precision of the computer that we're using. Okay, I think that's where I'm going to finish. We're just on time, are we? So there are a couple of issues here with these PWMs that one needs to be aware of, and some of which we'll already have noted. And we'll talk more about that, or I'll touch on this slide on Thursday, and then we'll talk about motif discovery, which is a lot more exciting, I think. So then we'll be using some cool algorithms, namely Gibbs sampling, in order to find the most enriched motifs in data sets. And this is a method that is generally applicable to many problems out there. So if you're interested in data mining, it's something to be aware of. So we'll talk more about that on Thursday. So I think I'll finish there. If you have any questions, let me know.